Okay, so we have Dr. George Daley joining us from Boston Children's Hospital, where he leads a stem cell institute, and we're grateful that he's here this morning to talk to us about consideration of potential epigenetic effects of mitochondrial replacement therapy.
Okay, thank you. I just want to indicate my conflicts. I am the director of the stem cell transplant program, which is primarily bone marrow transplant. We're quite actively engaged in somatic gene therapy at any given time. We have probably 2 or 3 kids on our service who are receiving this therapy, which now, after decades of investment, is really proving to be remarkably effective. My lab investigates the interface of stem cell biology and cancer. I am a consultant to MPM Capital, which is a diversified venture capital fund. My understanding is they don't have any direct interest in mitochondrial replacement approaches, but they have invested in companies where mitochondrial bioenergetics is relevant to the development of their products. I'm an officer of the International Society for Stem Cell Research. We are currently involved in revisions of international guidelines for human stem cell research and clinical translation in a committee that's chaired by Jonathan Kimmelman. Aspects of embryo research relevant to mitochondrial replacement are, in fact, also relevant to our discussions. We are primarily writing aspirational language. The committee has taken no stance on these deliberations, and I should add that Jonathan has been absolutely scrupulous in recusing himself from any discussions that might actually interfere or overlap with his work here for the IOM. I want to talk about a patient first, a patient that has influenced my thinking about mitochondrial disease. And this was a young girl that my fellow and I saw on rotation one time where we were called to the emergency room. This was a 3-year-old who had had transfusion-dependent anemia since birth, and a bone marrow biopsy at that early stage had shown ring sideroblasts, but really the diagnosis had been perplexing until, in fact, she presented on this occasion with a much more complex and obviously metabolic syndrome with diabetes, pancreatic insufficiency, and lactic acidosis, making us think about a mitochondrial disorder. And indeed, we took cells back to the lab. We're able to document a very significant 2.5 kb deletion, which knocked out some of the NADH dehydrogenases and a few of the tRNAs. We were also then able to say that this was, in fact, a condition of Pearson syndrome, which is something we do see on the hematology service because it often presents with a high degree of mitochondrial defects in the marrow and presents as anemia. So we took fibroblasts back to the laboratory and were able to derive induced pluripotent stem cells. Through this reprogramming process, we obviously then capture the defective mitochondria. And we could show by either southern blot or quantitative PCR a very high burden of defective mitochondrial heteroplasmy in our child. And we were able to then carry a number of clones over many passages in culture. And we were well aware of this notion of mitochondrial heteroplasmy and also of the descriptions in the medical literature of patients who had been transfusion dependent and later, in a sense, grown out of their condition, in part because of the segregation of healthier hematopoietic subclones and the ultimate clonal dominance of the hematopoietic system by these healthier subclones. And we, in fact, observed a very similar phenomenon among our induced pluripotent stem cells in culture. Here's an example of, just 3 of those over many passages. We had some here in blue that maintained this very high percentage of heteroplasmy, others which actually purged the defective mitochondria, ultimately leading to a cell line which was effectively isogenic but completely normalized in the mitochondrial pool, and then others that underwent this purging at a slightly later stage. So we were able to then do direct functional comparisons of these defective and purged cells, and under a number of different analyses, either the growth properties of the colonies, the mitochondrial membrane potential, or particularly relevant to this patient, for whom our concern was hematopoietic failure, we could demonstrate here in the hematopoietic colonies that were differentiated from the purged, normalized IPS cells, that these colonies tended to be more robust and larger than those that were derived from the lines that continued to maintain a very high degree of these defective mitochondria. We could also demonstrate the direct pathology of these sideroblasts, these abnormal red cell precursors, detected here as iron-laden particles in these paranuclear zones. In a blinded manner, our hematopathologist was actually able to tell that our purged cells had also been purged of these sideroblasts. So what this taught us was that reprogramming technology, Yamanaka-type IPS derivation, could in fact capture the mitochondrial pathology in these patient cells. We also made the very interesting observation that cell culture alone actually led to the normalization and purging, so that we could actually develop essentially healthy patient-specific cells. And that suggested to us that there might indeed be alternatives, cell-based types of therapies for individuals with this condition. And we were indeed considering the prospects of developing red cells as a transfusion strategy for this young girl. But very sadly, and I think emblematic of really the devastation of this kind of disease, our patient during that hospitalization ended up in the intensive care unit and ultimately succumbed to H1N1 flu. So these types of patients, I think, are illustrative of the tremendous burden of disease. And these was our paper that was published, and it was published alongside a paper from the Mayo Group that reported a very similar phenomenon in a patient with mitochondrial encephalopathy, lactic acidosis, and stroke syndrome, MELAS. So you've been talking about these papers, which are moving us ever closer to the realization of being able to perform spindle transfer and mitochondrial replacement in human embryos. What I want to talk about is what it would be a plausible strategy for performing as much quality control as might be possible within the limitations of current analytics. And I would say much of that would go along the following course. I've put together two of these illustrations from the web. Here's just the illustration of spindle transfer to produce a morula by mitochondrial replacement therapy, from which one could perform some type of blastomere biopsy. It's now well established that one can then cryopreserve the remaining embryo, and with a reasonable efficiency, probably approaching in some laboratories 50%, the ability to actually derive human embryonic stem cell lines, which can then be cultured and extensively characterized. And those characterizations I'll talk a little bit more about. So what are the prospects for understanding the genetic and epigenetic consequences of this at the preclinical level? I want to point out that my lab has been extensively involved in comparative genomics and epigenomics, addressing the question of how similar induced pluripotent stem cells, these reprogrammed skin cells, are to native embryo-derived embryonic stem cells. Obviously, this has a deep and fundamental scientific interest, but it's also been driven somewhat by the political debate as to whether or not there is IPS as a faithful alternative. And our group also published some of the earliest extensive comparisons of nuclear transfer reprogrammed embryo-derived cells and IPS cells and the like. So I've been quite sensitive to this issue, even though we have not done this in the context of any kind of mitochondrial replacement. But we do now have these IPS cells from patients with mitochondrial disease, and they exist alongside purged, essentially normalized variants. So these would, in fact, enable experiments which would allow us to define the links between the bioenergetic defects, and Vamsi could give you a wonderful lecture on how mitochondrial bioenergetics is now increasingly being understood to feed into various pools like acetyl-CoA and S-adenosylmethionine, and these have direct effects on chromatin-modifying enzymes. And so we're starting to see the connections between the metabolome of the cell, if you will, and the epigenome. And so we at least have a metric for performing those analyses. There's an increasing resolution, but still imperfect, of single-cell analytics. And obviously, one would have to really think in the early embryo, if you're going to do single blastomere biopsies, you really have to be very effective at single-cell analytics. And these are some of the strategies that are evolving. And then ultimately, with reasonable efficiency, one can derive embryonic stem cells from an embryo which would then be cryopreserved, allowing for a significant degree of both genetic analysis for copy number, whole genome sequencing, whole genome bisulfite sequencing, which allows you to capture the methylome, as well as differentiation analysis to be able to understand the effects on the segregation of perhaps carryover defective mitochondrial DNA into specific lineages and the like. And then, of course, allows correlations with organismal level analysis. And then let me just talk about the limitations, because it's absolutely clear that the single-cell analytics, while we're pushing the frontiers of this technology, are still quite limited. Our ability to sample transcripts for single-cell RNA-seq probably only gets to 20 to 30% of the expressed genome. We're much more limited in our ability to perform single-cell epigenomic analyses. We can't forget that there will be mosaicism among blastomeres, which once again limits our predictive ability. The cell we analyze, we can understand how close is it to the cells that we can't analyze. And there is always the potential that the selective process of in vitro culture plays an enormous role in the nature of the epigenome that we actually analyze. And we have now a much deeper understanding from comparative strategies between IPS, nuclear transfer ES, and fertilized embryo derivatives of the evolution of genotypes and epigenotypes. So I'm just going to leave it there, because I think we know a lot, but we always have to remain humble with how little we really do know and remain respectful of the unknown in these discussions. Thank you very much.
Thank you. So maybe we can get you both to sit at the front, and we can. I'll make a list.